Good evening, class. How are you, everyone? All good? How was the day? How was your day, everyone? Was it good? Hectic? A lot of schoolwork? Good. Mhm. Hectic. Mm, Sometime it is. For me, it was also a bit hectic, a bit down, but all in all, was productive. Your day needs to be productive, howsoever it is, that you get the maximum out of your day. At what time do you all wake up actually in the morning? And when do you go for sleep? You are having a student life, I know. It's very upside down. Might be sleeping late, waking up late then, and then slowly days getting off, especially during the lockdown times. Isn't it? The best time they say to be awake is 5 a.m. actually. Wake up at 5. There's a book by Robin Sharma. Uh, I had that book. It's called the 5 a.m. Club. Uh, 2 a.m. at night. Oh, 6 to 7. That's good. If you have that much power to do so, it's really good power. That's the highly intellectual and highly successful people timings are actually. If you are able to wake up at 6, sleeping by 1 or 2, and all day you are productive, that is quite refreshing and quite you can get a lot done in a day. Then a lot of things could be done if you are awake early in the morning, because there's a plenty of time to plan your day. What are the things that you have to do? What are the tasks that you have to achieve? What are the goals that were left behind, whether it's a personal goal or educational goal or intellectual goal or your hobbies or relationships or anything that has to be maintained. Um, yeah, during the early hours, so you make things done that these things has to be cleared, and then suck, you clear them out for the whole day. And the best part is you get a really sound sleep if you achieve your whole task. 2:30 a.m. What are you doing, guys, till 2 a.m.? That's very surprising. Studying whole night, or is it the phone thingy? That phone keeps you up busy till that time. Because that's very addictive. Nowadays, phones are really addictive. It's become part of our life. So let just few students to be add more. Um, then we can start. So today I will be dealing here in the lectures. So whatever we have done with the micropropagation, totopotency, morphogenesis, in vitro tissue culture, that the theoretical part that we did, uh, So take you long time. Whatever the theoretical part we did, that has to be done now practical point of view. How you could grow a rice grain with a micropropagation culture. How you could grow a mint plant from that. So what are the medium conditions that we required? What are the tissue culture things that we required? What are the equipments, instruments that we required? What kind of transplantation period we required? So all things matters. They matters a lot when you are working with tissue culture. So we will be dealing with the practical part of the tissue culture, though it's not feasible to take you to the lab. But with the videos that I have showed you on the first day with the tissue culture, I hope you have understood how that scientist did in the first video, and from the yesterday video that scientist did from two parts from the callus culture. So sorry, the organ culture and embryogenesis that he showed yesterday. So he showed how nicely it's done. And beyond that, that agrobacterium video that she has explained us how with the help of genetic engineering you could have a herbicide plant. So overall idea is to understand basic techniques of plant biotechnology. Maybe in the future you will be going for some practical aspects in your life. So for you people, these lectures that I'm gonna send you now, 5 lectures, so they will be really beneficial for you. Uh-huh. So can we add up everyday sprint assignment? So yeah, I mean it's not like you have to send me every day. You can send me at the end of your workshop. So just like day one, day two, day three, just create. You can create one whole document also. You can create different documents. It's up to you. It's just you should have a one file with you from this all these lectures. They are so that you can readily read them whenever you need of plant biotechnology. That's I give to everyone. So whenever you are going for any course, even in your class, in your subject, try to create from each day the summary out of it and try to write it in your notebook. So these things will be with you, so that you don't have to go through the lectures or the presentations at the time of need. So that's the main thing. Oh, that's long time to sleep, but we should instantly actually sleep. Otherwise, that's a kind of insomnia. It's called insomnia if you're sleeping quite, taking us long time. And it's just in this busy life. Earlier, the life was quite simple. In our days, we just quickly slang, just go down. But now things are like many things came up with the social media, which was not uh, feasible in our time. We don't have mobiles when we were studying. We don't have internet much of it, so we did everything on our own. So that's also one thing. For gate, um, I have to check. Maybe if you could share with me what is the syllabus for the gate, that would be beneficial for me. But what I'm teaching you here is, uh, oh, you hit the good point, Gaurav, meditation. Um, you hit the good point of whether this is feasible for GATE. So I have to check the syllabus, but the syllabus that I'm showing you is under the AIRI, Indian Agriculture Research Institute syllabus. So for the people who want to clear exam in their perspective in Indian agriculture aspect, so this is sufficient. This is more than sufficient if you cover all these topics. For the GATE, I am not sure. I have to check. Yeah, meditation. I started this meditation during my PhD time. Earlier, I never did that in my life. I was like, what is this voodoo science of sitting here and just checking out our breath or checking in between our eyes, third eye? So, but that's the key balance. If you understand to meditate yourself and channelize your chakras that we have behind, and just whether you do a meditation on some mantras or you do on your breathing, it doesn't matter, or just on your nose breathing, it should be just concentrate your whole energy on one thing and letting out your negativity out and let the positivity in. So this could be with any word, whether it's Buddhism, Sikhism, Hinduism, Muslims, or any Islamism. So any kind of religion, you just need to read through their stream, and then we are reaching to the uh, main path, main thing. That's a different path to go there. hmm So Telegram. I will check it later. So we had a lot of different talk today about meditation. We can share about our own experiences with meditation, how you could, like Sandeep Maheshwari, who was a kind of had seen a lot of failures in his life, but now he's the one of the India's number one motivational speaker, having a YouTube channel with golden card. So how he become it? It's basically he understood what is inside. He understood what is inside that will be the outside. So he cracked that point, and now he's helping everyone to go on top of their career, to solve their daily things, because it was all inside actually. uh, to all our questions, to all our misery, to all our happiness, to all our joy, to all our um, yeah, sorry to touch you these points. I should not go through this point. I should maybe concentrate on studies. So all the things that is the questions that are residing, uh, we try to read through the books, but in our Indian culture, they say that they always been inside. Just go inside, try to find it out. Where is the mess up thing? What are the blockages? Where is the free energy? How you could bring it out, and then concentrate on that, and then yeah, you can relieve from those things. Okay. uh, this we did yesterday. So for the student who are asking me about the gate, uh, so just want to share, like I got your, thank you for sharing this gate syllabus. So there is a plant and animal biotechnology. Within plant biotechnology, your transgenic plants will be covered, plant products of industrial importance, totipotency will be covered, regeneration of plants is covered, plant regulators is also somehow covered, tissue culture and cell suspension culture systems are covered. Uh, then under it, its nutrient kinetics methodology is almost covered. Production of secondary metabolites of plant suspension culture. So yeah, I mean it's more than enough. If you have gone through these syllabus, your most of the topics are covered actually. But still, I haven't cleared gate actually. I was always after my bachelor's, I was outside India, but I'm thinking of clearing it now actually. I've gone through its syllabus, and once I have cleared that, only then I can be sure, because things that you have never cleared and I start teaching them, that will be not good at all. So I will see. Uh, so how things are. Okay, lots of talk have we done today. Uh, um, yeah, what are the, so today I'm dealing with basically your practical part, that is a requirement from the plant tissue culture laboratory, techniques in plant tissue culture, media components and preparation will be done, and somehow we will do the micropropagation also. It's not written here. So mainly four parts will be covered today from this practical point of view. If we are still left, then maybe we can go for somoclonal variations to check somoclonal variants from our species from R0 to R1 to R2, getting a genotypically variant phenotype for our um, um, yeah, variable species. So we might go through that also today. So to start, uh, some this plant tissue. I will not repeat this one. This you have seen already. To how we aseptically done clear it, and we create that media in agar medium, cut the explant and grow them in the agar medium, and then at the end is a shoot formation, then the root formation as per the auxin and cytokinin ratio, and hence you have later on you move them from the agar medium to the um, yeah. So laboratory requirements. So what are the requirements to grow your plant tissue culture? Some basics are that it has to be aseptic, and the isolated plant part should be appropriately environment, which will help to divide the cells and to get the expression of your internal potential. So basic facilities that you required while doing in vitro process for the essential elements, you need a washing and storage facilities. That should be a separate room for that. And then a media preparation, sterilization and storage room. That should be a separate room for that also. Transfer area for aseptic manipulation, so where like laminar airflow where things will be happening for you. Culture rooms for incubators, maintenance of culture under controlled conditions, temperature, light, humidity. So a culture room should have a controlled conditions of temperature, light and humidity also. And one place would be your observational data where you will be observing the data. And the last and the sixth one is the transplantation area, that where you will be transplanting your plants, as in. So all these six are quite necessary places to run your lab. Otherwise, if one of these necessary departments are not available, then things could be really messed up. If things got messed up at any stage, so your whole tissue culture working, your whole money spent on media, or your time, everything will be wasted out. So things has to be, when you're working with the science, you have to be very serious, um, and it's not just, it's not fun. Sometimes it's fun, yes, but sometimes we have to be serious while doing so. So laboratory requirements. So we need a large sink area, running water in that. Drying ovens need to be there to autoclave our equipments like scalpels, uh, tweezers, all these equipment, scissors. Washing machine should be there to wash up our flask, clinical conical flask or any test tubes, to reuse them again and again. So steel buckets for soaking labware, acids or detergent baths, pipette washers, clinical brushes. So all these are quite required when you are need of washing and storage facilities. Then media preparation room. This is the central section of the laboratory where most of the activities are performed. That is media preparation and sterilization of media, and glassware is also needed for this culture. So this is quite necessary part also, media preparation. So following items are required in that room. Different types of glasswares, different kinds of balances, required chemicals, hot plates and stirrers, water bath, pH meter, autoclave hot air oven, microwave oven, vortex shaker, centrifuge, refrigerator, storage cabinet. I hope. Do you have any idea? Like, have you done these? Uh, have you used any of them in your practical experience with all these equipments? like centrifuge, vortex, microwave oven, autoclave, pH meter, water bath. Have you been used with them in regular use, students? Okay, so you know how a lab is working. So you know everything. So you also cell culture for several cell types. Okay, good, good, good. That's I need some assurance before I go ahead. So these are the basic things that we required before we start your plant tissue culture lab. Then you need a transfer area. Um, Tissue cultures can only be successful carried out in a very clean laboratory where you have protection against the airborne organisms. Laminar airflow is needed where you have a HEPA high efficiency particular air filter. Uh, then wiping flow with a 70% alcohol of your laminar airflow, and also putting up UV light which could kill the germicides inside it. And recently some people are started to have generate some germy air filters in which the air is passed through the high ultraviolet light and then giving us a light afterwards in the as air filters in our common rooms. So they are trying to bring this new technology also so that we have a virus free atmosphere in the room. So so this we are using for our lab chamber. Then comes your culture room, where you have to really take care of the temperature, light, photo period, humidity, air circulation, how it is. So all these things has to maintain very well there. And then further, culture rooms are constructed with the air condition has to be maintained so that what kind of temperature they required, everything is done preferably as per the plant's need. For suspension cultures, gyrator shakers are used, air conditioners and heaters are maintained at temperature of around 25 degrees Celsius, and humidity is maintained for the uniform force air ventilation. And lighting is done in the measure of foot candles, that is 40 to 200 FC in that case. Then data collection area has to be there where you're collecting your all data. This could be your, yeah, where you are that special facilities are there for them. So like growth and development of tissue culture in vitro are generally monitored by observing culture at the regular intervals, the culture room, and incubators where they have been maintained under controlled conditions. So these are the places where you can check your data collection area. Then after the data collection area, you have to transplant your area. So that is a transplantation area. So you transfer from the lab to the soil part like a greenhouse, where you have to acclimatize your plants first with the well humid conditions and control temperature and under control entry of sunlight. So you have to maintain everything quite nicely there also. So that's it. It was a just small review for you, like how you have to maintain for that uh, your the basic requirements for the plant tissue culture. Now, few slides about plant tissue culture. Before we start, just give me a second. Someone is calling me outside. Just give me 5 minutes. We start. Not 5 minutes, just 2 minutes, even less than 2 minutes. I'm coming in seconds. Apologize, students, for just few minutes. Someone was there. I have to take it in emergency. So anyhow, we are done with the basics requirements that are for the plant tissue culture laboratory. Now comes the uh, techniques in vitro in plant tissue culture. So what are the techniques that we use here? So mainly it's mainly four main steps. That is inoculation of X plant into your medium, then incubation of the culture, then after culture you do the subculturing to a different medium, then transplantation of the regenerated plants. That's how your main four steps for the tissue culture technique. That's the same video. Sorry, I think it's the same video. Yes. Um, then, to do so, to inoculate your X plant successfully, you have to take really precautions before you start at the time of transferring the sterilized X plants into the nutrient medium. Our dust, hair, hands, and clothes are the potential source of contamination, so these has to be taken care of. The hands should be wiped with a 95% alcohol, and the transfer area should be the clean and wiped with a 95% alcohol. Transfer process. Care should be taken so that the eggs plant not touch the edge of culture vessels, and cotton plug petri dishes to be sealed by the parafilms, and during the transfers also ensure that the plant tissue should be exposed to media properly. So all these things have to be taken care while you're doing your inoculation of X plant. Then comes the incubation. The incubation of the culture is done in the cultural room at 25 degrees Celsius. For some above or 2 degrees Celsius, and some tissues, some plants need even below or above 25 degrees Celsius as per the requirement. So some need a low light conditions for regeneration of light and dark periods are needed. And for the regeneration of plant well lighted, around 300 lux unit is required, and 16 hours light and 8 hours dark period is needed. That's how mostly in tissue culture there is a tube light is there with alarms, so they are like for 16 hours they will be on, and for the 8 hours they will be switched off. And also there's no light coming from the outside, so you maintain and you're providing with a refrigeration homogenization in that room where plants are growing, so you're maintaining everything while your incubation of culture is going on. Then low humidity causes the quick desiccation of culture medium, and high humidity favors the contamination of culture medium. More the water, then the contamination. If there's low humidity, then there might be your water might be removed from your culture medium. So specific relative humidity is required around 20 to 98% to maintain to done it properly. Then for cell suspension culture, agitation and aeration is secured by the use of shaker systems. Open platform orbital shakers are used for the same. Fluorescent lights are used to provide the day and night regimes to your culture. Then after incubation of a culture, then comes the subculturing of a culture, so that you have a very well defined uh, tissue culture without any contamination. So to do so, growth and development of tissues of culture in vitro are generally monitored and observe the culture of regular intervals in the culture of room. So you keep checking them how things are happening. So based on the observation with the hand or lens, simple microscope under aseptic condition, explants may be required to transfer to new medium with the new ingredients or hormone compositions depending upon the state of growth. So you remove that part from the previous culture to a new place with the new hormone composition, new ingredients, new media, and some precautions while you're doing so. Please take care as aseptic condition has to be there. So this aseptic condition will be keep on repeating every time, because once there's something goes wrong, then the whole process might go wrong. So at the end you might have contaminated seedlets, and hence your electricity, your time, your media, your cells, everything will be wasted out. So aseptic has to be maintained each aspect of your working out there. So in case of suspension culture, the change of media has to be done at quick intervals of times, and callus culture, so subculturing of callus tissue is needed to get the callus tissue in dividing conditions. Transplantation of regenerated plants. So plants regenerated from the in vitro tissue culture are transplanted to soil in pots. Prior to the transfer to the pots, acclimatization of these regenerated plants are needed. The plants at the time develop adequate roots and cuticular leaf surface so that it could withstand with the uh, field environment conditions. So process of acclimatization is done with the humid chamber so that the slowly your plant started to habitate the new high humid conditions to normal atmospheric humidity. Artificial light system is also required to include a mixture of fluorescent and incandescent lamps to provide balanced wavelength of light for the plant growth and photosynthesis. To beyond that, for the winter crops and summer crops, different temperature required. Air circulation is different, relative humidity is different. So as per the observation, we can see flowering and normal seed settings to get next progeny. So according to that, uh, your your actions will be happening then. Then that's it for this one also. Uh, okay, so we are done with the basic part of tissue culture. So now these lectures are from the uh, Punjab Agriculture University. So I will be sharing with you these parts as the PDF. So I was teaching the same in my college time. So they were quite handy and to the point, and felt like sharing with you all also. So they will come with the all the calculation part, how things are done in practically. If you have got a lab there, you can also go and start one of these experiments whenever you got the time. So you can also say to your plant biotechnology professor in your college, if you're working under in future you might have one semester for it. You will come overcome by these kind of experiments for sure. So so as we are done so far with the basic presentations, now comes the practical parts because first I want to just let you have a flash of how tissue culture is done, what are the main requirements. Now requirements are done, now comes the practical part. So this one is the same like orientation to the tissue culture and genetic transformation facility. So as we have discussed already, you need a washing room. So this is quite practical. This is what you really need when you are working there in the lab. So all these things that we have discussed so far. So single span of room is needed, glasswares, double sink, wall racks, ovens. Then for the media preparation room, that is your media kitchen, we can say that it should be around 12 by 13-inch room. So that these should include your all oven, microwave, refrigerator, trolleys, water bath, racks, spatulas, weighing balance, pH meter, double sink, water purification units, autoclave. So just think, guys. So establishing a lab is not an easy task. You need so many things, small things, to make it a complete lab. So keep your wherever if you're working in the lab, worship your lab. Actually, I have to suggest because I believe in work is worship. Whatever you're working, that should be your worship actually, and you're working really hard for the for experiments or your results. So in future, if you got a chance to do to go for PhD or your master thesis, so keep respect of each things that yet you're working in the lab. So because it's yeah. So then comes your transfer room as we discussed, which are requiring for the safety burners, bead sterilizers, binocular stemma microscopes, cultural trolleys, incubators. So all these things are required in that case also. So further in that, then incubation room is there. The incubation room will be like a culture room where your growth room of is about 22 to by 18 feet inches inches are required. It should be maintained by 25 degrees Celsius. We have discussed all these. 30 to 5000 lux has to be maintained. 16 by 8 hours light has to be there, and relative humidity should be 60 to 80% as per the requirement. It should have air conditioner, heat blowers, shakers, wall thermometers, humidifiers, timers to regulate. So yeah, then contains the laboratory for if you have a genetic transformation laboratory, so for them you need agrobacterium tumefaciens for sure, or particle gun method could be used. Then you need that machine for particle gun method, and further you need laminar airflow, tabletop centrifuge, helium gas cylinders, PCR machines, electrophoresis apparatus, vacuum pump, sonicator, particle gun, refrigerator, vortexer, micropipette. So these are basic requirements uh, of your genetic transformation laboratory. Then for the molecular biology lab, you need all these 22 equipments, because molecular biology lab has a lot of experiments you can manage in that. So to do so, you need from pH meter to hot plate stirrer, PCR machines, weighing balance, deep freezers for -20 to -70 degrees Celsius. So all these things are quite important while doing experiments. Then there should be a cold room where it is maintained at 4 degrees Celsius for required for DNA protein work, because some of your DNA protein they work at you have to preserve them or shake them at 4 degrees Celsius. So a room is also required as a cold room for experiments. Then comes the dark room. Dark room should be equipped with cameras, is required for the photography. So you have to take some images for your results. Then it's also required. Then comes the storeroom. Then greenhouse. All in all, then glass house and transgenic glass house. So as per the requirement, we need all these 7, 8, 9, 10, 11, 12 things to grow your tissue culture lab. So it seems like a whole big project whenever you're working. I had experience of working in my PhD, but my work was into the animal side. Once my master thesis was on the metastasis, cancer metastasis, and my PhD was onto the cardiovascular physiology of the heart, how you um, kill how could the antioxidants could help to overcome the heart attack. So mine was totally into the medicinal field, but my master's was actually agrobiotechnology. So there we were dealing with a lot of plant breeding part, gene gun method. We use a lot of time. Agrobacterium tumefaciens, genetical engineering we did quite number of times. Also we had a lot of biochemistry techniques in the plant tissue culture, and also high next generation sequencing NGS was being highly used at that time, and we were working on the canola, that is canola oil, the raps oil, the rap seeds. So raps flour we were working on that time to create a new variety of that. And there the first time it was in 2011 actually. Drones were used to click the pictures. So I was in this my master's and both PhD was in Germany. So I was seeing at that time that drones were used to click pictures from the top. But that was were quite an unsuccessful experiments. But now they are coming by Amazon to drop our things, our delivery. So that is future is quite near now with us. So that was something about the laboratory, the what things that you required to do your daily routine experiments, and each thing is interdependent on each other. If something is not working, so the whole experiments could be kaput, will be bad then. So maintain that thing. Then comes the if you're having a plant tissue culture, you're growing your um, you started to start your experiments, so you required a plant medium, a culture media, which is quite aseptic and full of nutrients, from the macronutrients to micronutrients, vitamins, minerals, and all kind of uh, energetic nutrients that require for the tissue culture. So so we will discuss about this now in the second part of these experiments, that is plant cell culture and media preparations. So, in media is used in plant tissue culture is composed of essential elements, that is macro and micro organic supplements like vitamins and amino acids, sucrose, growth regulators, and water. So all these, yeah, together essential elements, organic supplements, sucrose, growth regulators, and water. These five elements together make your a nice media, a healthy media. So these are required for in vitro growth of the tissue. Therefore, successful plant tissue or tissue culture is dependent upon the composition of the nutrient media used. Um, so these are the mainly the macronutrients milligram per liter and micronutrients milligram per liter. So NH4NO3, NH4H2PO4, KNO3, CaCaCl2, 2H2O, MgS4, 7H2O, KH2PO4, NH4 2SO4, NH2PO4, H2O. So all these things are required for the micronutrients. And if you're growing MS media, B5 media, so there are four types of media, different types of media for different conditions are required. So MS media, B5 media, WH media, DKW media. So all these different media will have a different uh, composition of nutrients then. So we have divided them into very nicely in these tables. So if you want to grow MS media, then you need NH4NO3 to be 1650 milligram per liter. Then further KNO3 to be 1900. Then respectively calcium chloride 2.2H2O to be 440. Then 370 magnesium sulfate. Then potassium hydrogen phosphate to be 170. So like this, you have to maintain. Um, your micronutrients. Then comes organic, like nicotinic acid, paradoxin HCl, thymosin HCl, glycine, minoinositol. So all these things are required as per the uh, the, the medium that you're making. Now for carbohydrate, you need sucrose to be added 30 milligram, 30,000 milligram per liter. Uh, and then so so the macronutrients basically includes your nitrogen, phosphorus, potassium, magnesium, calcium, sulfur, carbon, hydrogen, and oxygen. They are the various microelements, and they mainly comprise of 0%, 0.1% of the dry weight of the plants. That is very very low percentage, 0.1% of your whole dry weight plants. Then the micronutrients, these includes our like manganese, iodine, copper, cobalt, boron, molybdenum, iron, zinc. They are all comprises of your micronutrient. Whereas ethylene diamine tetracetic acid EDTA is used in conjunction with the iron sulfate also. So EDTA complex with iron so as to allow slow and continuous release of iron into the medium. So that's an uncomplexed iron can precipitate out of the media as a ferric acid also, ferric oxide also. So on organic supplements, you have vitamin B1, that is thiamine, is considered essential for culture of plant in vitro, whereas amino acids provide a source of reduced nitrogen. So most frequently is glycine is being used. Glycine, remember that. Then comes the carbohydrates, and carbohydrates are cheap and easily most readily assimilated. Relatively stable one is the sucrose in the carbohydrates. So other carbohydrates such as glucose, maltose, galactose, and sorbitose could also be used in that case. So to prepare, so that was to show you like what are the various five basic medias, nutrients, microorganic, carbohydrates are required. So oh, now comes to prepare if you are preparing a MS media, that is this one, the first one, how you have how you can Murshige and Skoog medium, very famous one, MS media. Though it's nowadays it's not required in the lab to prepare, but sometimes you are asked to prepare media for yourself. So you should be ready for that also. But we can get these media readily prepared from the market, and it's also not so expensive. It's quite cheap. So that's one of the beauty of working with the Murshiga Skoog medium. So the most common used medium plant in plant tissue culture is the Murshigan Skoog medium. So first we prepared a stock solution, so which is given in table 2. So these are the various. First we created 20x, 50x, 50x, 100x, 100x of these solutions in 2 liters. Then out of these solutions, we take 50 ml out of this, 20 ml, 20 ml, 10 ml, and 10 ml. So that's how you have to prepare your first MS media, which was done in 1962. So you take a cylinder in which you have 800 ml of distilled water, and then you add stocks like from the stock 150 ml, then stock 220 ml, then 20 ml, 10 ml, 10 ml. Then add sucrose, 30 gram of sucrose, and mannitol as 100 milligram to the above stock and dissolve by stirring. Then add rest of the water to make it to the 1000 ml. Then you have to adjust the pH to 5.8 by using pH meter, adding sodium hydroxide or HCl drop wise. Then place the medium on the hot plate and warm it. To this add 7.5 gram of agar medium and dissolve it, heating the medium. Then you dispense the medium in test tube or jars, autoclave at 121 degrees Celsius at 15 psi for 20 to 25 minutes. So this is done. Your media is prepared here like this. These are told you some little bit like dissolved calcium, copper sulfate, sodium monoxide. So how you have to do it with them. So then you autoclave your media at 121 degrees Celsius at 15 psi pressure, and then autoclaving is done mainly for 20 to 25 minutes. So then minimum autoclaving time is done. So for plant tissue culture medium, so as per the volume, for if your volume is 25 ml, so minimum autoclaving time is 20 minutes. So as the ml is increasing, so if it is a 2 liter of your medium, then you need to autoclave for 48 degrees Celsius. So some medium components are thermoliable, that is they could be destroyed at high temperature, so should not be autoclaved. Just take care of that while doing so. So then afterwards, your autoclaving is done. Then the solidification of media is done. Then if there is a browning is happening in your plants because of the phenolic compounds of the plant tissue culture, so anti-browning substance has to be added also in the media, like citric acid, ascorbic acid, activated charcoals, polyvinyl pyridine, or something like these has to be added into that. So take care of that also. So that's it. That was something about the media. um, um, so here we can take a 10 minutes break now, and then we start with the next part of your meristematic tip culture and production of disease plants. Okay, then just take a break. Then we see us at somehow around 9 o'clock or so. So we deal with this part also, and then we do one more thing also for today. Okay, see you soon. So, class, I'm back. Um, so, let's start with the third experiment. We are done with the basic and then medium preparation. Now comes the meristematic dip culture for production of disease-free plants. So, in this, basically, you are growing a meristematic mentha plant with the help of the meristematic tissue, meristematic tip. In this, what you do at the first step is you cut the shoot apices from the in vitro growth shoot culture of mentha, then excise aseptically meristem measures using stereo microscope in the petri dish and put in the laminar airflow. Then you culture the meristem in the test tube containing about 10 ml semi-solid MS medium. Supplementary with indole acetic acid, carnitine, incubate for 3 to 4 weeks in good light intensity. Then comes your induction of rooting meristematic drive plants, culturing onto the half strength basal medium, MS basal medium, then hardening of your meristematic drive plants, then transfer to the soil. So this is how it is showing that if you got a chance to do practical, you have a plant, then you take the meristem and you check them under the stereo microscope under the autoclave conditions, that it should look like this. Yeah. Then what you do is you culture them on the medium with the culture on the medium with the meristem inside the test tube. You plug it and you incubate for 3 weeks, then selenate out of it. Then you transfer to the rooting media, incubate for 2 weeks. Then you add to the rooting medium. Then you have roots started to grow. Then you remove the virus. If you check the virus indexing by a ELISA test or so, then if everything is fine, then you put into the hardening and transfer to the soil. That is your virus-free plant will be produced at the end. So percent of plant regeneration you can check is number of meristem forming plants in your test tube by total number of meristem cultured. So this will be your percentage of plant regeneration multiplied by 100. And the virus indexing could be done using indicator plants or using a ELISA test kit. So you can do by using both of any test to check your whether you have a virus-free plant or not. So that was something to do with the mint plant that you can do the meristem culture with that. Then comes the establishment of callus and cell suspension culture. So in this callus and cell suspension culture, basically main objective is to demonstrate your total potency of the induction of plant regeneration from such cultures, to induce homoclonal variations, to isolate protoplasts, to develop somatic embryos, production of secondary metabolites, genetic transformation of plants. So all these things could be taken care while you're dealing with the callus culture and cell suspension culture. So in this case, we are doing just simple callus culture from the rice part of rice grain. So what you do, you take a healthy mature rice grain and you sterilize with 0.1% of mercury chloride for 10 minutes. Then you wash the grain with sterile water in the laminar airflow. Then you inoculate them aseptically with MS media 5 to 10 gram per culture vessel, medium supplements with carnitine and 2,4-D, then incubate the culture in total dark in growth room for 3 to 4 weeks. So that's the procedure for your growing your rice culture as in callus culture if you want callus out of it. So percentage to calculate the percentage of callusing, number of grains you have exhibited by total number showing callus formation divided by the total number of grains cultured. So you can calculate the percentage of callusing, multiply by 100. So observation has to be taken for the callus initiation, callus growth, color, texture of callus under the stereo microscope. Then you keep checking how its color, everything, morphological patterns, how things are working with it. Further establishment of cell suspension culture in rice. Uh, procedure is take 1 to 2 gram of actively growing callus, friable callus, then macerate it aseptically in 7 to 10 ml of liquid AA medium. That is this one is the medium that in so next part what you do is the with the help of AA medium you start the next step with it. First you done is callousing is done. Now next part is establishment of cell culture in rice. So this is it is done. So you have a rice grain, you culture in a callus induction medium. So you have callus induction is done. And so after 10 days you have subculturing of it. Then after subculturing you have callus, you have medium. So after 1 month transfer callus to the cell suspension liquid medium on shaker. Then you have a callus growing on it. Then frequent subculturing, callus break down to the aggregates of free cells of single cells. Then you have a cell suspension after 1 month. So this is how you establish your callus and cell suspension culture. This is all shown here. You can calculate the growth rate also. Also the percentage of dead cells by adding even blue, 2% of even blue into the solution. So after 5 minutes you will see a blue stained cells out of them, and hence you can calculate by number of blue dead cells by total number of cells in the microscopic field. So that's it from this part. So this all you can this you can, if you like, you can make a small, you have to make one small yeah assignment out of it, 2 page summary of plant tissue culture. So all these topics are covered now, so I will stop it here, and then I would like to take you to the DNA extraction. Yeah, how DNA extraction is done. So any questions so far? It was quite straightforward. This is like techniques that I was telling you today. Any questions so far, बच्चों? Shall we continue with the next technique? Okay, I think you all slept now. You're all sleeping. So I have decided today. Yes, go ahead. Yes, yes, go ahead. So I was teaching you today all the uh, so today I will be taking care of the mainly the practical part, so we will finish with it today. So now I want to take it to the demonstration of isolation of DNA, how you can isolate DNA, the sixth part, and the gel electrophoresis technique. So we will do these two in virtual lab, so which will be very interactive and good to know. So please wakey, wake up and concentrate on the things. So DNA extraction virtual lab. Let's start the lab. So in this experimental, we need to isolate DNA from human test subject. So instead of, let's say in this we are doing it from the human, but for example, let's say we are isolating from the plants also. So we just need plant samples in that case, then we can do it. So just think that the source is plant, but all the technique is same. Is all the technique is same. So DNA isolation could be done to check various things actually. for genetic testing, body identification, analysis of forensic evidence. In our case, so in plant, also in the plant case of in plant transgenic plants, to check how it is going on. So we can do in that case also DNA isolation. So DNA isolation is typically the first step in longer laboratory process. So let's start with the cell. So this is your cell. Then you have your nucleus inside it. Then inside the nucleus is a chromosomes lying there. In the chromosomes, then you have your histones. Then histones are connected with the DNA, and and we need to collect this DNA. So where we first have to collect cells from the test subject. So here we are taking the cells from the test subject as a swab. This is we are also doing in the COVID test nowadays. We take from the mouth and also from the nose. Skins from the, in this case we are taking swab cells, but in the COVID test they take it from the inside, somewhere from the thorax. So let's get started. So let's first will we check the collect the cheek cells, then we burst cells upon to release DNA, then we separate DNA from protein and debris, then we isolate concentrated DNA. So these are the all things that we required while isolating the DNA. Warm water bath, centrifuge, buccal swab, micropipettes, sample tubes, lysis solution, concentrated salt solution, resuspension buffer, ethanol, isopropyl alcohol. So we need all these mediums to isolate our DNA. So first from buccal swab, we remove some cells and put in the Eppendorf. So here is our cheek cells with the nucleus. Inside the nucleus is DNA is present. So we have our lysis solution to remove the cell out or the outer cell wall to separate things from each other. So lysis means to separate or to break down, sorry. And after the lysis solution in our DNA, we put them into the water bath. So lysis solution will do, it will open up the nucleus, open up the chromosome, destroy the histones where our DNA is coiled around it with the help of protein kinase K. So now we are adding a salt solution to it, to the tube. So salt solution will separate DNA from the debris, protein and debris. So they will clump together. Place the tube in the centrifuge and try to balance with another centrifuge on the other side and turn on it. So you have protein debris and DNA liquid. So inside the centrifuge, your debris will be down, and in the liquid side will be your all DNA liquid at the end. So we take the DNA liquid and put it up there into a new Eppendorf. And to that we add isopropyl alcohol now. So here, after inverting the tube several times, we have an isopropyl alcohol is mixed with the DNA solution. So DNA is not soluble in this isopropyl alcohol, so it will come out of the solution. You can see clumped DNA of naked eye. So this will not mix in that, so it's become a clumped, and we can see with the naked eye this DNA. So we put this tube into the Eppendorf or into microcentrifuge, then close it, run it. Take the supernatant, and then once the liquid is removed, DNA is allowed to dry. So or everything down is in the pallet is your DNA, and you can store it in the freezer for many years, or you can use it in the next experiments. So you have just extracted DNA. So that was quite easy to do. So just it's just adding A plus B plus C and D, and then hence you have your DNA in your hands. So that was the virtual lab for the DNA isolation of DNA. Once you have isolated DNA, now you can do the gel electrophoresis. Gel electrophoresis is done to check how much DNA is produced in your samples. What is the mega base pairs, how much mega base pairs of DNA is being produced. So to calculate that, we use gel electrophoresis as a technique. Let's start with that now. As in it's a virtual lab of the same. So this will be now your actually last experiment from this. Rest we have done already in the theory so much. So demonstration of gel electrophoresis technique. So So after the isolation of DNA, now comes the next part of gel electrophoresis. So here we are holding a small tube with some clear liquid in it, and it's a DNA of several different lengths. So it has several different lengths of DNA, and we have to get to know which amount of DNA we have. Our job is to require to know understand the length of our DNA. So how we will do it? We will do it with the help of gel electrophoresis. So like this, let's say in the previous virtual lab, we have short, medium and long DNA with different strand size. So you can see we have triple in the long, medium here and short here. So both, but DNA strands are molecules so tiny that you can't see them under your most microscopes. So is there a way to measure this without microscope? Yes, by gel electrophoresis. So scientists are able to understand its length with the help of this technique, and they can separate their molecules like proteins also in this case. So how does it work actually? What is the basic principle behind it? So this is your gel, and you can see small pores in it. So gel has little sponge made of gellio pores in it with small holes in it. So once you add your DNA molecule, once we place DNA sample into holes at one end of the gel, one side we made it negative and positive. As your DNA is negatively charged because of the phosphate groups, it will move in this direction from negative to positive. So DNA started to move in those from the small holes. So the one which are small, they will travel faster more. The one which are big, they will travel slower and they will remain at the top. And this is how you can differentiate between different DNA samples with different DNA strands. So in this case, mainly three are present in this two, small and middle, and this small and bigger, and middle and longer are present. So in each human, different DNAs are present. So under the UV light, you can see the stained how your DNA bands looks like afterwards. So this is DNA ladder, which is telling us the how much size is of your DNA, and you can check the DNA. Uh, yeah, DNA size of your working. So now let's we have understood the basic principle now. Now it comes to understand its basic working. So these are the steps. First we will make the gel, then the setup of gel apparatus, load the DNA sample into the gel, hook up the electrical current and run the gel, then stain up the gel and analyze the results. So these are the various steps we will do. So you need agarose, flask, gel mold, gel combs, buffer, microwave. So all these things are required for the case of gel electrophoresis. So you took small amount of seaweed agarose and put into the conical flask and add some water buffer, liquid buffer to the flask which has electrical charges. Then you put into the microwave. So it will boil inside that. It will become in the liquid state and then you put into your gel chamber. Okay. Then you take a gel comb uh, and you put on your gel chamber to create those holes that we require to run our gel electrophoresis. Then gel gets solidified and we remove this gel chamber out of it, gel combs. Now we put them into the electrophoretic box. So first we add the buffer, then we add this into inside of this, then we add our samples to it. So we will add our loading buffer, DNA sample, DNA size standard. So first thing first, uh, we add loading buffer to our DNA sample. So this will help to load our gel. Otherwise, it's very little, not heavy. It will might float in the medium. So it will help to make it to the color blue also that our samples are running, and it will also help make it heavy. So take it and pour it here into your first chamber. Inject it slowly. You go inside, and that's it. So in real life, it looks quite easy, but In real life, no, sorry, here it looks like it quite easy, but in real life it's quite difficult because mostly you pour your everything out. So we take some DNA size standard and we pour it that also because it sometimes come come into the other well also. This should not happen. Then you collect the red. So yeah, black with the black, red with the red. Then you turn on the power plug. So our gel is running. If you see these bubbles, tiny air bubbles going on, that means our gel is running. So this is how your gel is going on. So take the gel mold, and so we will add into ethidium bromide, which will attach to the base pairs of our DNA, and we can see our gel very nicely into that. Yeah. So I will put this into ethidium bromide and check for, we put them in for half an hour and then check under ultra UV light. Remove it and turn it on, and this is how it looks like. So this was your DNA standards, right? This was our DNA. So this will be how much size? Around, this will be how much, and this will be how much? So I think this will be around 1500, and this will be around 3500. This will be around 6000. So all your estimations are correct. So this is how, this is the DNA ladder, and we have calculated how much DNA is there. So there was 1500 base pairs, there was 3500 base pairs, and 6000 base pairs of DNA. And we have calculated with the help of this technique. So DNA isolated, we can calculate how much DNA was there. So that's it for today, I guess. We have done quite number of techniques today, practical parts, some tissue techniques, some isolation of DNA, some of the yeah. So so that's it for today, class. And okay, there's a question. Before the incubation of X plant, there's a mentioning of 95% uses of ethanol instead of 75%. How can we use 95 as bacterial proteins get coagulated in this case, which ultimately create resistance for appropriate sterilization to sterilization system step? Uh, so where was it? In the techniques, we have discussed this. So बच्चे, this is just बिल्कुल at the starting of your working, because we haven't started to work, and at the 95% alcohol transfer area will be very clean and wipe with the 95% and transfer process, and all this talking and sneezing should be avoided there. So that is okay. I mean, mostly we use 70% there, but 95% they have told us to do it in the case of tissue culture. In our case, we were also using 75% ethanol, and we were using also other chemicals to protect our samples from mycoplasma. Some scientists were really crazy to do so. So 95% is too much actually. So 75% is wise to use it. This can kill most of the microorganisms that are not required for experiments. So any questions so far? I think I will deal with the next slides of your subjective slides tomorrow about uh, this one, somoclanification and somatic embryogenesis in synthetic seeds. So we will do this tomorrow about this. So today we are done with the practical part very nicely, and then we continue series wise as it is. So we will manage it very easily, so no worries. Okay then. Uh, is it everyone is there? Could you give me hands up, please? Thumbs up, please, if you're there. It feels like I'm talking to the ghost or talking to myself so far. Okay, okay, okay. So my voice comes late to you, then you reply. So it's a lagging period. Yes, yes, I will share these gel electrophoresis and DNA isolation. So I can share it now with you. Oopsie. Just the links of everything. So okay guys, I will end the stream. Thank you very much for today. Oh, there is one question. Please give one example where stage 2 medium is being used for stage 3 medium as well. Okay. Uh, I have stage 2. But yeah, mediums are used normally for the all the stages. But sometimes we change them as per the needs are. So that has to be with specific conditions. Um, let's say if we are growing rose flower, we take X plant from the rose flower, and throughout the whole stage we have to keep one medium only. So let's say MS media, that is mostly quietly used. So if it is required to change the media, basically we will change its various hormones like auxin or cytokinin, or it needs some extra hormones, we change according to that. So I cannot specifically say that from this media to this media in that stage we change it. Yeah. So if you know some answer, please let me know also. But as per in details, I'm not sure of it so far. Okay, if you have any other thing, then I will say good night, take care, sleep well, stay safe, and please like, subscribe, and share of this YouTube channel so that I could be motivated. Please like these videos. This really motivates me to keep making new content for you and keep generating new stuff for you. So maybe I will start some other classes also on YouTube, but at the moment I'm just concentrated for you students for these classes. So I'm not creating any content further. So I'm just trying to make it. You can give me some ideas what kind of content you need maybe later, so I can create some content on YouTube channel for you all. So about that we will talk about this tomorrow. Give me your ideas, keep it with you. And meantime, sleep well then again. Good night. Bye bye. Take care.